Hey guys, what's new? Welcome back. Quackers here, as you can tell. And today we're going to be talking about something kinda interesting. Not the wristband. That's pretty cool too, from my sister. But no, today we're going to be talking about something even more interesting. We're going to be talking about protein synthesis the Quackers way. Alright, so what is protein synthesis? Protein synthesis is the process by which organisms make proteins. It's one way where genotype controls phenotype. Kinda interesting, kinda not. I'm not sure. Don't quote me on that. Ms. Forgosh, don't get angry. Alright, so gene expression. Gene expression is the process of converting genotype to phenotype. Gene expression is basically where you're expressing the gene, hence gene expression. Because the gene, that physical makeup is the phenotype. The actual makeup on the inside, that's the genotype. Alright, so what are the steps of protein synthesis? I'm sure these thoughts are boggling your mind right now, and there's only two pretty simple steps. The first step, transcription. That's the process of going from DNA to RNA. DNA cannot be removed, I repeat, cannot be removed from the nucleus. So they make a photocopy in the form of RNA. Step 2, translation. The process of going from RNA to proteins. The photocopy is then used to make the recipe. I get it. It's like a cookbook. You see, I'm sure you do. Alright, so I'm sure you're interested. What the heck am I talking about, Quackers? Well, young lads, I'm sure that you'll be glad to know that I can answer that question. Transcription. DNA once again, is transcribed, aka copied, to make a complementary RNA sequence. It's similar to DNA replication. RNA polymerase will look at the DNA sequence and make a complementary RNA sequence using a DNA template. These are a lot of big words. I'll get to them later. But just so you know, RNA polymerase, that adds nucleotide sequences to only one strand, and it's involved in protein synthesis. Synthesis. God damn it, Quacker. Just get it through your thick skull. Alright, so, RNA polymerase. RNA, single-stranded. DNA, double-stranded. Deoxyribonucleic acid, ribonucleic acid. Simple enough, yes. I'm sure it is for any of you who took school or even science class at any point in their lives. Alright, so, transcription. There's 3 steps apparently. I was wrong. Sometimes we get wrong things, kids. That's how you do it on the Quacker Show. But you know what? You learn from those mistakes. So, 3 steps. Initiation. The RNA polymerase binds to the DNA. It binds to the DNA and begins separating strands. Step 2, elongation. RNA polymerase adds ribonucleotides complementary to the DNA template. That's kind of interesting. It's kind of like they smushed together, made a baby or something, right? So, termination. That's the 3rd step. Termination. Ending, pretty hype. So, termination. RNA transcript, the RNA copy of the DNA, is released, separated from the DNA strands. My sister's right back here. She's going on YouTube. She wants to be famous and whatnot.
Who are you talking to?
We're talking to absolutely nobody. We're just learning about DNA protein synthesis! Woo! Baby, yeah! So, termination. Let's get back on subject. Termination. The RNA transcript is released. Separated DNA strands come together, and RNA polymerase leaves. Okay, so there's a few types of RNA. Kind of interesting, isn't it? So, the first type, mRNA, messenger RNA, is transcribed instructions for making proteins, the RNA transcript. 2, ribosomal RNA, rRNA, like but different, So, ribosomal RNA. RNA within ribosomes is the stuff that's in ribosomes. Simple? Yes. Awesome. So, 3, transfer RNA, tRNA. It brings amino acids, which are the building blocks of proteins, to the ribosomes. That's pretty hype, isn't it? Quackers would love to say so. So, translation. When the RNA transcript is used to determine the amino acid sequence to make proteins. Pretty hype, methinks. Okay, so let's get a little bit back on track. So, codons. the sequence of 3 nucleotide bases that act as words and each correspond with a specific amino acid. Just like AUG is an AUG. It's like that thing that go brr in CSGO, but different, you know? Awesome. So, AUG, as a matter of fact, is actually the start codon. Now, there's a few stop codons, but AUG is the only start codon, according to my notes at any rate, and my notes are pretty detailed. They're not just scribbles, I promise you. So, anyways, AUG, that's the start codon. A few of the stop codons are UAA, UAG, and UGA. Those are just a couple of them, and by a couple of them, I mean all of them. So, translation. It occurs in ribosomes. Pretty cool. It occurs in the cytoplasm specifically of rough ER. So, my notes are currently telling me that the anti-codon is complementary to the codon sequence on the mRNA transcript, which codes for the specific amino acid. So, anti-codons, they're essentially the opposite, but not really, you know? I hope you know, because I don't really. I mean, I totally know. Quackers is very confident, as you can tell. Okay, so 3 steps of translation, just like 3 steps of transcription. Pretty cool, pretty amazing, kind of crazy. I know. I'm shaking right now with how insane that is. Gosh, I can't just help but feel like there's somebody behind me, but there's nobody there. Do you guys know? Is it that big subscribe button that you should be pressing? I'm just joking with you. Don't press it. It's not cool. So, the 3 steps of translation at any rate. First, initiation is the 2 ribosomal subunits that bind to the mRNA transcript, along with the first tRNA. Pretty snazzy notes. 2. Elongation. Amino acids are added to the growing polypeptide chain. Amino acids are brought by tRNA to the ribosome based on their complementary anticodon region. They connect to each other by peptide bonds, not hydrogen bonds, peptide bonds, not polypeptide bonds. Gosh, Quackers, just get it right. Alright, the 3rd step, termination. Stop. When the stop codon is reached, ribosomal subunits separate. mRNA transcript is released along with the new synthesized polypeptide chain. Proteins. Amazing. Hype. So crazy, am I right? Alrighty here. Because this video isn't scuffed enough, we're just going to continue. Alright, I think we got everything. So, guys, leave any questions you have down below in the comment section. Don't forget to hit that red sub button rather smoothly, if I'm not ahead. That like button, don't click. You click that like button, you're not my friend anymore, okay? Alright, chill. So, I hope you guys enjoyed the video. Thank you so much for watching. It was by far one of the stupidest videos I've ever done, but I love doing this anyways because it's grades and I need them. Comment. Quackers needs an A if you got this far. Alright, thank you so much for watching, everybody. It's been real with you. Quackers is out. Ciao.